Hello and welcome. I'm Stuart Wolpert. We have a terrific program for you today. Dr. Ming Guo is a physician and a scientist with both an MD and a PhD who treats patients with neurological disorders. Dr. Guo holds UCLA's Laurie and Stephen C. Gordon Chair in Neurosciences. She's a professor of neurology, molecular, and medical pharmacology at David Geffen's School of Medicine at UCLA. Dr. Guo is also director of UCLA's Asian Center. She's won major awards from the Alfred P. Sloan Foundation, the National Institutes of Health, the McKnight Foundation, many others. If I told you about all of Dr. Guo's awards and honors, we'd run out of time before she has a chance to say anything to you. So, with no further ado, please join me in welcoming Dr. Ming Guo.
Thank you so much, Stuart. Thank you so much for your invitation. And I know we usually say right now the 80s is new, is new 60s. This is the middle age. So I really enjoy seeing patients in the middle age. So delighted to be here. As Stuart said, I'm a physician scientist, seeing patients with brain degeneration. I also run a research lab investigating the causes and potential finding therapies for these devastating illnesses.
So Dr. Guo, your research focuses on learning the causes of brain and muscle diseases of aging, discovering their molecular basis, and even finding cures if possible for diseases that are currently incurable. And you also treat patients who come from Europe, Asia, India, of course the United States. You came to the United States from China with very little money, but with dreams and ambitions. So before we get into any of the rest of it, the research and all the wonderful work that you do, please tell us about your journey as both a doctor who cares for patients and a scientist conducting research on brain aging.
Yeah, I'd be happy to share about my journey. I was born in China during the Cultural Revolution. My parents were both sent to the countryside to be re-educated because they were intellectuals, they were college professors. I only met my father when I was 3. Rather, my father got the chance to meet me at the age of 3 because I had no recollection. And later, the family reunited. And initially, I wanted to go to study engineers because my family members are all engineers, so it is expected I would be an engineer. However, during high school, I watched a soap opera made by Japan, and then there was this teenager girl who developed leukemia. Everyone in the family is trying to save her. That really formed a very deep, had really a deep impression on me. Then I thought, what would be more impactful to save someone's life? So I decided to go to medical school. So during medical school, I was very motivated and thinking that in the end I would help treating all these patients. Then I realized there's a lot of diseases we really have no idea how to treat. That prompted me to decided to pursue a research, ultimately decided to apply for graduate school in the United States. I got accepted. However, I came here with only $30 in my pocket.
Wow.
Yeah, and my parents spent half of their lifelong savings to buy a one-way ticket for me to come here. Wow. So then I was determined I had to make it. So I went to UC San Francisco to study research. Initially I wanted to do pain control. Obviously, as a physician you can understand this is something very important. Then I realized there are new discoveries of molecular biology which would open up a new front for developing new therapies. So, and ultimately pursued a PhD on molecular biology. However, I never really forgot what initially motivated me to help patients, then I decided to do a residency at UCLA in neurology. During the residency in neurology, I encountered numerous patients and their families who have neurological diseases, particularly a group of disorders called neurodegenerative diseases, including Alzheimer's, Parkinson's, among many others. Together, there is still no cure for these disorders. And then with the aging population, these group of disorders is going to be increasing. So I decided to pursue a joint research and patient care career on this group of disorders known as neurodegenerative diseases.
Before we get into the research, what was your life like when you first came here? Did you know people here? Did you speak much English?
Well, I did learn English. As a matter of fact, my medical education was in English with English textbooks. So, but my English was not very helpful in the sense that it's not American English. What I was learned was, how do you do? Then if you say, thank you very much, I would say, not at all. That was the English I learned. So when I first came and they said, thank you very much, you're welcome. Like, you're welcome, what is that? So I had to relearn everything. Yeah.
Did you know people?
I only know very few, yeah, but largely, yeah. I was the only one here.
Okay, so how does your research inform your treatment of patients, and how does your treatment of patients impact your research?
It's a great question. So obviously, patient provides a constant source of motivation for someone who is also interested in finding cures for these incurable diseases. And obviously, so this is cannot be underestimated. Another aspect. So this is really a two-directional interactions. On the other hand, by telling patients and their families that I'm really researching and studying the group of diseases that you have provides enormous hope for the family.
That's good, because I was wondering if it might be frustrating for you. These patients are coming great distances to come see you, and these diseases really can't be cured.
Completely. It is a sometimes frustrating process. I would rather tell the patient, look, here's the pill, and then take it, everything will be fine. But sometimes part of the discussions will be, this is currently what happens, and we don't really have a cure, but nonetheless, there are certain treatment, there's some management. You also provide a kind of guide through the family, because this is a long process. And family is really a unit rather than the patient itself, himself or herself.
So you're director of UCLA's Aging Center. Your laboratory focuses on identifying potential therapeutic targets for brain and muscle diseases. Can you tell us a bit about the center's mission?
The center's mission is, in a way, trying to understand aging, ultimately reverse aging. And then later I'll be able to provide you a little bit more information on that, particularly our impact. To have, like the slideshow, to live longer and better relies on us to have brain health and health of many organs, and also to reduce a group of disorders known as disease of aging, which includes cancer, heart disease, diabetes, stroke, neurodegenerative disease, and many others.
Wow, your goal is to reverse aging. I mean, that's remarkable. And we understand the brain the least compared with the heart and other organs, so that's quite an ambitious goal. To change that. Now, one thing I've heard you say is that Parkinson's is not one disease; it's many diseases. I've heard that about cancer, but I hadn't heard that before about Parkinson's. Tell us about Parkinson's.
Yeah, so I study a group of neurodegenerative disease. Perhaps I can move on to a little bit. So a group of diseases known as neurodegenerative disorders, which include Alzheimer's, Parkinson's, ALS, frontotemporal dementia, and many others. So they affect really more than at least a third of us, if not half of us, and then there's no cure for these diseases, and we can't stop the progression of this disease. So my lab over the years have been focused on Alzheimer's disease, Parkinson's disease, frontotemporal dementia, and the sporadic forms of ALS. So as Stuart mentioned, if we take Parkinson's, for example, it's not one disease. It's a syndrome with many different causes. To understand each individual causes and ultimately identify a targeted or precise cure would be very meaningful.
So your laboratory was the first in the world to discover that two Parkinson's disease genes work together to control the quality and health of mitochondria. Now, I might say that when I was a student, I never realized the importance of mitochondria, and now I see that our mitochondria is really vital to our health. So we will be talking for a bit about mitochondria, but about these two genes that work together that you discovered. Tell us about how these disease genes work together to control the quality and health of mitochondria, and about how defects in mitochondrial function are an important cause of Parkinson's disease.
Sure, let me show you some visuals. So as Stuart mentioned, I study Parkinson's disease among other diseases with an emphasis on mitochondria. Now, what is mitochondria? Mitochondria is part of the cell, what we call organelle of the cell, that really is illustrated here on the left-hand side. It is a powerhouse of the cell, provide much of the energy. The simple way of thinking about this is it is like batteries that provides a currency, cellular currency, energy currency known as ATP. These, of course, mitochondria take on oxygen and food and generates energy. Mitochondria also has its own DNA, and damaged mitochondria, as well as mitochondria DNA, accumulate with aging and many diseases. So you want me to talk a little bit about Parkinson's disease, these two genes?
Yeah, sure. But also, these two genes are called PIG1 and Parkin, which are mutated in some people with Parkinson's. So tell us about Parkinson's and the two genes.
Yeah. We've emphasis on a group of Parkinson's disease genes, which means an inherited form of Parkinson's disease. Mutations in a handful of genes right now, the list is expanding, including PINK1 and Parkin, cause inherited forms of Parkinson's disease. Perhaps our best known work in my laboratory with my team members were to identify the function of PINK1 and to find out that PINK1 and Parkin function together as partners to control mitochondrial integrity and quality. So this provides significant insight for the field. Number one, it says mitochondria dysfunction is an important causal factors for causing at least part of Parkinson's disease. Number two, mitochondrial quality, it is really important because there are two of these genes actually function together in a common molecular pathway. In future, if we target this pathway, there's a chance we can find the cure and help patients with these genetic forms of mutations as well as patients that currently have no genetic forms of these, no genetic defect on these genes.
So mitochondria often become damaged with age. So as people age, the damaged mitochondria tend to accumulate in the brain, in the muscles, and other organs. And when cells can't eliminate damaged mitochondria, those mitochondria can become toxic. They contribute to a wide range of diseases that you're studying, that you're treating. So what's known about the role of these two genes, PINK1 and Parkin, in Parkinson's disease? And how are these two genes linked to the disease?
Yeah, mutation and dysfunction of these genes leads to Parkinson's disease. And the process is known as mitochondrial quality control. What does that mean? As we age, the quality of mitochondria, which are largely, each cell can have hundreds, if not thousands of mitochondria that provides energy. As we age, these mitochondria, individual mitochondria, and together as a population-based accumulate damage. So once the damage reach a certain threshold, these cells would have dysfunction and ultimate death. So that's the foundation basis for generating Parkinson's disease. Since we identified this important pathway that controls mitochondrial quality, we were surprised to find, this is right now, thousands of labs in the worldwide are looking at this pathway. It turns out the dysfunction of PINK1 and Parkin not only can impact Parkinson's disease, but also some other neurodegenerative diseases, cancer, diabetes, heart disease, and many others.
Wow. So these two genes are implicated in all of those, cancer, heart disease, diabetes?
Yeah, so we initially found out the function of PINK1, and also most importantly, we found these two genes function as partners to control mitochondrial integrity and quality. As you could imagine, to maintain a high quality of mitochondria are not only important in Parkinson's disease, you can imagine it may impact some other diseases as well.
So mitochondrial DNA gets damaged as we get older. Can you tell us about the importance of how a cell disposes of unnecessary and damaged components? So there's a Greek word called autophagy, which means self-eating. And does the cell destroy these defective components? Does the cell recycle the components into other cellular parts? And the cell does so less effectively as we get older, right?
Yes, good question. As we know, mitochondria, I like to think about mitochondria as in a certain way like a nuclear power plant, because it's so efficient in generating energy. But on the other hand, no one in their right mind would choose to live next to a power plant, nuclear power plant, right here. Because the reason is if there's ever a leakage, that would do enormous damage to surrounding neighborhood. Mitochondria is the same thing. As mitochondria reach a reduced quality, then it potentially could release all these oxidative damages, among others, that poison the surrounding vicinity of the cellular component. If this happens, as an example is a nuclear power plant, we need to really contain and control this leakage. So the way to do it is to contain. In the cellular basis, in a cell, what we do is a process called mitophagy. What does that mean? It means mitochondrial autophagy. Then what is autophagy? You probably have read some of the sort of public science articles on autophagy. Auto is self, phagy, meaning eating yourself. That sounds pretty morbid, but what happens is within the cell, if you identify a component of the cell that you think is not the best, this time mitochondria, what you do is you mobilize the cellular processes, which this time is mitophagy, and to just recycle this part of mitochondria. So this is an important, very important process, and it turns out the key component for mitophagy is the pink-Parkin pathway.
Oh, I want to hear more about that. So are PINK1 and Parkin important in eliminating the damaged mitochondrial DNA that accumulate?
Yeah, it is. It's important in eliminating mitochondria as well as mitochondrial DNA. So yes, it is. That's why this is the key component of that process.
And you're saying these two genes, PINK1 and Parkin, work together to control the quality of mitochondria, but by recycling or discarding damaged mitochondria?
Exactly right.
Wow. And how do they do that?
What they do is, interestingly, our body naturally have PINK1 and Parkin. When there's a damage, the mitochondria would have, so it has a layer that has sort of potential, energy potential, and when that energy potential changes, it will stabilize the protein PINK1, and PINK1 would send a signal to the rest of the cell looking for Parkin. This is sort of like SOS signal. Then Parkin would then move on to this damaged mitochondria, then ultimately together they elicit this process and recruit autophagosomes, then facilitate this mitochondria autophagy.
And what happens to the damaged mitochondria? Does it get recycled and then used
Yeah.
in cellular parts?
Yeah, so when it's entered into this compartment called autophagosomes, and these components are perfectly useful, that they just disintegrated and recycled for components. This is sort of a process like if we ever have a certain shortage or problematic, one room is not good, then we recycle the component to the room and to be used to fuel other aspects of cellular function.
So each cell has hundreds of mitochondria or even thousands of mitochondria, I think. In your lab, are you able to reverse damage to mitochondrial DNA?
Yeah, so let me just show you. I'll tell you first with respect to whole mitochondria in the context of the disease. Then I'll tell you about the process related to single mitochondria, related to mitochondrial DNA. So as I told you, we've uncovered that PINK1 and Parkin are a key component of this process that controls mitochondria quality and integrity. Over the years, through laboratory studies, we are looking for ways to reverse these mutation phenotypes. In essence, we generate animal models that mimic the Parkinson's disease. Then we asked, what are the things we can do to reverse these pathology such that these will be basis for developing therapies? Over the years, we have identified several genes that when we manipulate these genes, they can cause the pathology to go away or at least significantly reduced.
Wow,
some of these components are listed here.
Wow, remarkable. You can reverse the damage. Wow.
Yeah, and then most importantly, not only the cellular damage, the pathologies are also reversed.
Well, you've done that in your laboratory. That's
Yes,
remarkable.
yes.
So you conduct research with fruit flies as well as mice and tissue culture cells, and 75% of human disease genes have have counterparts in fruit flies. So I know sometimes people may wonder, why is a fruit fly a model organism that researchers use? But like humans, the flies age, they learn, they can get cancer, they have memory, they have immunity, they have circadian rhythm. How confident are you that your findings with fruit flies and with other organisms apply in humans?
We're very confident. When people ask these questions, and you among others, particularly my patient, every time I said we're studying Alzheimer's and Parkinson's among other brain degenerative diseases, they usually say, great, so faster. So the next, they will be say, fruit flies, why? This is a common question. It turns out from genetic makeup standpoint, fruit flies are really not that different from us. The building blocks are very similar. So if you look at certain behaviors like memory and certain behaviors like even cancer, immunity, even addiction are shared between us. Rather, I would say the genes control these processes are shared among humans and fruit flies.
Wow, fruit flies can be addicted.
Yes, exactly. 75% of the human disease genes have Drosophila, have fruit fly counterpart. From that standpoint, we as human beings are not really that different from fruit flies. And there are 13 Nobel Prize winners are actually studying fruit flies. There is also circadian rhythm, among others.
I hope you'll be the 14th.
Yeah, so it is really interesting when I also tell people that, so also when you study fruit flies and you don't have many like animal activists lining up outside of your laboratory to protest. However, usually after some talks, people in the audience would say, "Doctor, don't say that. You just told me they can have memory, they have circadian rhythm, they can sleep, they can have many different features that we commonly have. Perhaps in future they will line up outside of your laboratory." Yeah, not yet.
I know the patients are lining up to see you.
Yeah.
So have you removed damaged mitochondrial DNA in fruit flies?
Yes, so
Wow.
perhaps that comes to the second theme in my lab. Just overall, my lab have two main themes. There are many things we study, but one theme, the whole theme is about aging and neurodegenerative diseases. One is to target these brain degenerative diseases directly, such that we study Parkinson's disease. We also, over the years, studied another group of diseases that cause frontotemporal dementia and muscle degeneration. And we actually identified compounds that can reverse that pathology in both animals and human patient cells. And then another theme that we're having is to potentially reverse aging. Why do we want to do that? Then we can move on to talk a little bit about aging. Can anybody here tell me, what do you think is the oldest individuals thus far in this world?
I know the answer to that. Older.
What did you
She said 106.
106,
That's too
okay.
young. That's too young.
What else? Yes. You are absolutely correct. So here's the slides. The longest living human being thus far is a French lady. She lived 122.5. So obviously people were fascinated about her. And then, as you can see, this record, she died in 1997. This record hasn't been broken after, almost 30 years. So people were fascinated. Always says, what did she do? What did she do in the past, in her life? What did she do that I'm not doing, right? So it's an obvious question. It turns out she sold pencils to Van Gogh, think about the time, and then she took up fencing at the age of 85. So that says great, even if any age is good to start with anything. So she rode bicycles till 100 years old. And last one, though, she quit smoking at the age of 119. So then the question is, people says, do you think we should, maybe that's the secret of longevity, maybe I should start smoking? The answer is no. So, and then there's all these anecdotes about her. For example, there were all these, every time after she was 100 years old, every year there are all these reporters lining up to interview her, just to see her. Then they always says, there was one reporter said, I hope that we'll be able to see you next year. And she said, young man, there's no reason you can't make it next year. So this is fantastic. So talking about, of course, aging is always important, and particularly in the context of diseases. So I told you there's the group of disorders that we talked about, known as disease of aging, which include heart disease, and heart disease, cancer, stroke, diabetes, and neurodegenerative disease, such as Alzheimer's disease, among others. So if you look at this chart, this really says what our whole overarching goal. On the x-axis, this says timing, as we were born and as we age. On the y-axis is the incidence of age-related diseases. As we age, the incidence of these age-related disease keeps going up, and the pace is really accelerating. Our goal is to be able to pull down this aging curve such that in the end we were able to say at the given age, meaning at the given time point, our incidence of these diseases will be reduced. So what is the point if we live longer but we're demented, right? This is not our goal. So this is the motivation to study age-related diseases.
That's such important work. I have many more questions for you, but I do want to pause for a moment to say that that's just a remarkably ambitious goal. Your research agenda, your work with patients is just fabulous. We're very fortunate to have you here. So I think in a lot of the cases, when you go up to the top of that, people's mitochondria gets damaged, and then it becomes toxic, that causes diseases. Although also, I know you were telling me the other day that there are cases where children who normally would have healthy mitochondria, cases where they don't, and where they have really devastating diseases at young ages, and I guess that would be kind of the lower end, where a small number of young people do have these serious diseases, and again, maybe related to damaged mitochondria. Is that right?
Yeah, so the previous slides, what I'm showing is the y-axis is the incidence of age-related disease, but your question is really related to what kind of diseases or processes that accumulated mitochondria, damaged mitochondria DNA would cause. Let me just first step back to show you this chart. So on the top right-hand side, that's a cell. So our individuals, each one of us have numerous cells in our body. And then if you look at each of the individual cells, there's this component known as mitochondria, and which is amplified here. And mitochondria also contains mitochondrial DNA. As you know, we heard about DNA, which is the genetic materials, but most of the time when we talk about mitochondria, when we talk about DNA, that's related to nuclear DNA. However, there's only one other part of the cell that human cells have DNA, which is mitochondria DNA, which we abbreviated as mtDNA. Here, if you look at the mitochondria, then it's amplified to these ring-shaped DNA. It's known as mitochondrial DNA. This is the key site of aging. Mitochondrial DNA accumulate damage as we age, resulting in cellular dysfunction and death. So, as Stuart mentioned, damaged mitochondrial DNA leads to pathology in the brain, muscle, heart, and pancreas, illustrated here. And our goal is really to remove damaged mitochondrial DNA in tissues to reverse aging.
Wow, and how's that going?
That goes very well. When we collaborated with researchers from Caltech, which are a bunch of genius, so to generate a new tool which was not present before we started the process, to generate transgenic animals that contain damaged mitochondria. So as we know, generating mutations in the nuclear genome is trivial nowadays due to technologies as CRISPRs and among others. However, generating mutations in mitochondrial DNA, it was very challenging because, as you can imagine, think about the process. If one cell that has hundreds, if not thousands of mitochondria, and each mitochondria has this numerous copies of mitochondrial DNA, and then to generate mutation on each part of them is quite challenging technologically. But nonetheless, we prevailed. We generated mutation. But most importantly, we found that by certain processes, one of the processes, our favorite gene, PINK1 and Parkin, we can remove up to 95% of damaged mitochondrial DNA.
Wow.
So this was in the context of the animals, but right now, of course, we set up more collaborations with collaborators here at UCLA and at Caltech. So we're very passionate into finding out the similar processes in human cells. Ultimately, this would be the fountain of youth.
Wow.
This is related
That sounds like it's worth the 14th Nobel Prize.
So, great. We should recommend you to be on the committee. So, as Stuart also mentioned, about accumulated damaged mitochondrial DNA can also cause a group of disorders starting from childhood, and some of these patients can live into the adulthood. This group of diseases is known as mitochondrial diseases. So this group of diseases, they can have preferentially affecting the brain, the muscle, your nervous system, including your vision and your eyesight, and pancreas, among others. Individual children usually would get epilepsy, mental retardation, and weakness of muscle. Sometimes they need ventilator to help them to breathe, and hearing loss and vision loss and diabetes, among others. So if we can find these compounds that can, in humans, right now we found it in animals, that we will be able to not only provide fountain of youth for all of us here, including myself, as well as for effective treatment for these children.
Wow,
Yeah.
that's really remarkable. So if I understand correctly, for most of your career you were focused on looking for insights into Parkinson's disease and Alzheimer's. But are you saying that PINK1, these two genes that work together, PINK1 and Parkin, that they're also implicated not only in Parkinson's and Alzheimer's, but maybe in these other, I think what you call diseases of aging, so cancers, heart disease, diabetes. I mean, might those genes and mitochondria kind of be a key to learning about a lot of these diseases that we're all susceptible to?
Yes, very much so. When we started the process, we were already excited to find out these genes, when mutated, cause familial Parkinson's disease, really work together to control mitochondria, to make mitochondria healthier and higher quality. So we were very excited about that, but little did we know that over the years right now, more than 1,000 labs worldwide have studied many different processes. We only studied a small part of it, but the rest of the world cumulatively provide multiple evidences suggesting these genes are also involved in heart disease, diabetes, cancer, even immunity, lung disease, many others. So it is, yeah.
That doesn't sound to me like you're only studying a small part.
Yeah, yeah. So it is important to think, if you think about as a biologist, we obviously found out these foundational pieces of biology. Not only did we, so I like to think my lab works on two processes. One is really related to disease, right? We found the causes of Parkinson's disease, and we found genes then when we changing of these genes can reverse the pathology. But at the same time, we also uncovered a process that previously were not known to biologists, known as maintaining mitochondrial quality. If you think about that, it is not surprising, right? For many diseases in our heart, in our other cells, to maintain mitochondrial quality should be very important. When this process breaks down, one might actually develop some other diseases. So therefore, our studies on PINK1 Parkin can provide multiple insight into these other diseases as well.
So Parkinson's disease affects, I think, 5% of people over age 80, and 70% of Parkinson's patients develop cognitive decline. Alzheimer's afflicts half of all people over 85. By 2050, I think 16 million Americans are projected to have Alzheimer's. So these advances that you're making toward understanding Alzheimer's and Parkinson's and possible new treatments are just really critical. Congratulations.
Yeah, so because I'm thinking I would possibly get the disease, so I better work hard on that. My mother, yeah, let me just show you some slides of my mother. So,
Chemistry professor.
This is my mother, and this is me. She's right now aged 91. While this picture is taken, she was 81. So it's not quite 91.
Looks younger.
Yeah, so she is a chemistry professor. She visited me here, and we really enjoyed spending time together. My mother always tell me one thing. I'm not afraid of dying, because this is particularly pertinent when we celebrated her 90th birthday. She made sure that she told me this. She's a tiger mom, by the way you want to know. Tiger mom meaning she really tells you what to do. Even if I'm a professor here, she tells me what to do every day. So she told me that she's not afraid of dying. This is an inevitable path of life, but she doesn't want to develop certain diseases. One disease is dementia. which she doesn't want to lose her memory, and then this is not the life she wanted to live. Another thing, of course, she doesn't want to be paralyzed. She says, you work harder to reverse aging, so to make me at least 5 years younger. So what is the impact of 5 years younger? Because for Alzheimer's disease, every 5 years after a certain age, say age 65, our chances of developing Alzheimer's disease doubles. So that's very meaningful. She said, right now I have a perfectly good mind, but do you think I can be free from dementia? Then I said, Mom, I wish so, but I have to tell you, no. Every 5 years, there's a chance to develop Alzheimer's. The chance of developing Alzheimer's disease would be double. So that's why she says, if we could pause the aging, we could potentially cut the global impact of Alzheimer's disease by half. That's why we need to work hard. So may I just, following up, since we're here on the theme of mom, so that here my mom not only tells me what to do, she also helped me to do this work. This again is a slide when she visited me, 15 years earlier. So she is not a biology professor. She's not a biologist. What she is she visited me as a retired chemistry professor, but she is very interested in helping me in whatever way she could. As you know, as a Chinese mom, they cook you dinner, do your laundry, does everything, clean your household. But at night, when I went back home, I wanted to go to the lab because there's so many things I wanted to do. And I kind of felt guilty of leaving her at home, but then ultimately we decided we could just all go to the lab, working together. This is what we sit side by side doing fruit fly work. I'll tell you what it is. And this is also, people say, when you study fruit flies, wouldn't they fly away? So she had several questions. She said, I don't really like flies. They look ugly. But when I showed her what fruit flies look like, she said, oh, they look quite nice, because it's completely different when you look at a housefly. Anyways, so what we do here is we put flies in these pads. So then we pass carbon dioxide, CO2, so the fly would be temporarily anesthetized, so to speak. Then you can put on the pad, then you can use an art brush to flip them, look around, look at them under the microscope, which is here. Afterwards, you said, okay, I sorted out my flies, then you can put it back into these either vials or these bottles, then fly recover. So then you can study them some other time. So it is very interesting to know, this is different from chemistry experiments, so you may have an idea, because when I'm doing experiment like chemistry or molecular biology, I have to concentrate. If I'm really thinking about something else, if I'm mixed up, then I'm like, which tube did I just add? However, when you do fruit flies research, you don't need to have that high level of concentration. So you just look at them and you said, okay, this one, then you can talk. So I concluded that if you want to bond with your parents, working on fruit flies is one of the best ways.
Well, that's wonderful. And if your mother is watching the video of this program, I want to say congratulations on being such an inspiring role model to Dr. Guo. But,
yes. And as a tiger mom.
Okay. Yeah.
You work harder.
But as I was writing questions for today's program, there were 3 questions that stood out that I was really eager to ask you. So here's one of them, the 2 others are coming a little bit later. So you talked about the French woman who's 122 and a half. Can aging be delayed by altering the function of a gene or a group of genes, or are aging and decline inevitable?
Well, obviously, our work, at least in animals, have shown we can remove the significant aging signatures in the animal model. And the idea is, if you believe that our building block through evolution, and then whatever mechanism that is, that our building block is the same, then you would imagine if we could pause or reverse aging, that this would be the same for human beings.
Wow.
Yeah, even some other
Reverse
researchers,
aging. Wow.
yes. Or pause or delay. All of them I'd be happy about.
Slow down aging.
Yeah, so if I can pause, let's just say delayed, ideally pause at this moment,
Wow.
I'd be very happy.
That's
I think you would be happy too.
Yeah.
Yeah.
So a gene called MOL1 suppresses a PINK1 mutant. How important is MOL1? I know that's something that you've studied about. How important is MOL1 in mitochondrial health? And is MOL1 a potential genetic target for treating Parkinson's disease?
Yeah, so if we look at the slides, we
Oh,
have, yeah, so we have identified, this is all the work from my lab. So we found that mutations of PINK1 and Parkin work together as partners to maintain our mitochondrial quality and integrity. And then we, over the years, have identified all these genes, and more are coming. When mutated, MOL1 is there, so that can stop the pathology and make us healthier. So MOL1 is one of them. It is very important because if we provide this gene product, we're gonna stop, reduce pathology of PINK1 and Parkin. So this potentially would be a very important therapeutic target. So not only that, as you can see, there are multiple other entry points that one can develop therapies. So that we're very excited, more are coming.
So you have several genetic targets. MOL1 is one. You have several.
Yeah, so as you can see, MOL1, mitofuscin, BCPP97, and DRP1, and clueless. In fly genes, we have these wonderful names, right? Clueless, don't you just love that? So initially we were clueless about the function, but later we identified some clues how they function to help mitochondrial quality.
Is there anything you want to say about neuroinflammation and whether your research addresses the relationship between neuroinflammation and diseases like Parkinson's?
Neuroinflammation is one of the key components right now for neurodegenerative diseases. So more and more evidence suggests this is an important process. Our work per se has not addressed neuroinflammation. However, I wanted to tell you, neuroinflammation also has a mitochondrial component that we're currently studying.
So from everything you've said, you've convinced me that my wife and I should go to a spa and buy a mitochondrial cleanse. Unfortunately, there's no mitochondrial cleanse, at least not yet. So in terms of what we can do, does your research provide evidence, insights into the role that diet and exercise play in brain and muscle diseases? So for example, does regular exercise help to keep our mitochondria healthy until we can get that mitochondrial cleanse?
Yeah, so your work really mentioned two aspects. One is what I'd love to envision about future mitochondria spa. This is doesn't have to be only limited for women. I know women love spa, but men as well. So what we do is perhaps periodically we can go to a mitochondria spa, cleanse our damaged mitochondria, and then we would be rejuvenated, or rather our mitochondria are rejuvenated, so our cells are rejuvenated. Therefore, we might be healthier, in a more healthier state. The second question you asked is something to do with what we can do to make us healthier. So yes, what you said is all true. Diet, exercise. By the way, one of the key components of exercise is acting on mitochondria. Of course, sleep is very important. Again, there's a component of mitochondria and stress reduction, and among others.
Well, let's go back to the first part of that. Do you think we might see a mitochondrial spa in your lifetime?
Yes. Yes, in my lifetime, yes. But although I need to work harder, take into my mother's comments every day.
That's remarkable. It's remarkable. And you're saying that exercise does improve the health of mitochondria?
Yes.
And would that even include walking, or that would be more?
Well, so there are different aspects of exercise, and then there's different group of literature suggesting multiple different things, but overall, exercise have that increase of certain hormones in your brain and also improve your mitochondria. And then, by the way, mitochondria has other impact. For example, mental health component has also a mitochondria component, meaning if you improve mitochondria function, your mental health is likely to be improved as well.
One other question. When we talk about Alzheimer's, dementia, Lewy body dementia, are there differences? These are all different disorders, or are
Great question. Great question. I frequently am asked these questions a lot, and people ask, what is really dementia? What is really Alzheimer's? Are they the same thing or different things? So what is dementia? Dementia is a dysfunction, cognitive impairment of your brain, which can include memory loss, language deficit, visual spatial skills, personality changes, among others. So that to meet the criteria of dementia, you also need to, these cognitive dysfunctions also need to impact your activities of daily living. So this group, as you know, dementia is quite broad. It can have more than 100 different causes. There's reversible causes of dementia. There's also potentially irreversible causes of dementia. So then among dementia category, Alzheimer's disease is one part of it. So in a way, dementia is the umbrella term. Alzheimer's disease is part of the dementia. So there are what are the other causes of dementia? Of course, vascular dementia. If you get certain vascular lesions, including stroke, then you can potentially get dementia. Parkinson's disease. As a matter of fact, 70% individuals with Parkinson's disease can have cognitive impairment, some of them dementia. And then, as you mentioned, there's another group of dementia known as Lewy body dementia, which is the second most common neurodegenerative dementia. And of course, we studied another dementia in my lab, known as also frontal temporal dementia, which has a different presentation, preferentially impact your language and behaviors. So those are different kind of dementias.
So if it weren't enough that you're doing all this research in your laboratory, you're treating patients, you're serving on editorial boards and giving lectures and things. I know you also work with partners in industry, including a startup company, to help develop new treatments, and scientists can't do it by themselves. Industrial partners are important. Tell us a little bit about these partnerships.
Yeah, when I was younger, I was quite ambitious. I thought I would make a discovery, and then I will control, find the cures of the diseases and so on. Then as I matured a little bit, I realized I can't do it myself. So, and also if our goals are in the field to develop cures for these devastating illnesses, so we need to get people together to partner. So in that context, I serve as a member of the board of directors of an organization known as American Neurological Association, which is a professional organization for neurologists, which is more clinical discipline, as well as neuroscientists, which are more research research area. So what I initiated a new initiative as a board member is to establish an industry and academia partnership. So letting companies and academia world to talk to each other. We previously had a little bit of discussion, but this needs to be a whole lot more to facilitate or accelerated the process of finding cure. In that context, I'm a member of the scientific board at UCLA, a place at the CNSI called Magnify, which is a group of startup companies that are located in-house at UCLA to facilitate also the process of developing new treatment.
That's the California Nanosystems Institute.
Yes.
Another great example
Yes.
of work you're doing. Okay, so one of the other questions that I really wanted to ask you. So now that you've learned all of this about mitochondria, Ming Guo, Parkinson, and MOL1, and these diseases of aging, what advice do you have for us about maintaining good health, keeping mitochondria healthy? You know, I've heard you say before that the following things are all helpful: plant-based diet, Mediterranean diet, moderate physical exercise, increased mental activities, don't smoke, unlike the 122-year-old, positive social and family interactions, that they're all important for good health. But tell us a little bit more, maybe are there things that you told your mother to do, or that you're doing, or specific pieces of advice that can keep us all healthier?
Yeah, there are many pieces of this. Of course, diet right now, the certain evidences are on perhaps Mediterranean diet, although there are other type of diet that people are studying. And exercise, moderate is in a way good, but increase certain intensity, such as to make our cardiovascular fitness stronger, potentially can have a stronger impact to protect our health span. Because as I said, longevity, lifespan is one part of it, but really health span is very important. What is the point of just having a health span without lifespan, right? This is what my mother drilled down to me every day.
The other way around. The point of lifespan.
Lifespan is the point of health span is more important.
Yes.
Yes. So, and of course, sleep is critically important, as we recently identified that during sleep, that our brain has a natural process to remove toxic compound in our brain, which can potentially lead to Alzheimer's disease. And stress is very important. So if we have positive stress, it's not all bad stress. I mean, it's not all stress is bad. Certain stress is good to your body, but not chronically bad stress. And there are many other. Social interaction is very important. Like we're sitting here, this is wonderful. We're learning something. We meet friends together, so make us healthier.
We're learning a lot. And so what kinds of exercise is it? Walking, riding a bicycle, lifting weights?
So again, this is a broad field. There are certain exercise, such as aerobic exercise, there are resistance or strength training, there are all this intensity training, there's also zone 2 training. All carries a certain different kind of aspect of health. If you want to do, you have a combination of all of that, that would be fantastic as well.
Those are my questions. Now let's hear your questions.
Mindfulness have been good to reduce stress. Some studies have not been shown that, when I'm talking about, yes, by common sense that should be helpful, but I think it may be helpful to indirectly affecting sleep, there are certain studies, like even clinical trials in saying mindfulness, that hasn't been shown as a very strong evidence for dementia yet. But in general, that could improve the quality of sleep if done right. Yes.
Our last program was about mindfulness with Professor Sarah Melzer, and the video of that is available. Yes.
Wait for the microphone, please.
I can project. I have a question about hereditary. Can you give us any insight on how hereditary affects these degenerative diseases?
Yeah, it's a great question. Hereditary, as we know, for Alzheimer's disease, there are certain genes, when mutated, can lead to very bad Alzheimer's disease. The bad, I mean very early onset. So these onset of Alzheimer's in general for individuals will be after the age of 65. For a patient with 3 potential genes, these age can be, the age of onset can be significantly earlier. For example, at the age of 30, to the age of 50s. And the youngest patient I've seen is age of 27. Imagine that. So there is actually a good movie and a book called Still Alice. How many of them have read about it? A few of them, yeah, great. It's a fantastic book portraying a Harvard professor who's brilliant, of course. And interestingly, she's a linguistic professor, always very proud of her language skills. Just prior to the age of 50, she noticed, in the movie you would see that she was actually giving a lecture at UCLA. So then all of a sudden she had difficulty coming up with words. This was very surprising to her. And one thing led to another, that she realized she had this familial forms of Alzheimer's disease. And let's talk about second common cause of neurodegenerative disease, which is Parkinson's disease. So mutations of probably right now a little less than 10 genes, including PINK1 and PARKIN, can cause 15% of Parkinson's disease. Of course, we in my lab studied PINK1, PARKIN, and other work I haven't told you about of other genes. This is also very important to lead to Parkinson's disease. We also worked on in my lab a frontal dementia, which is also a genetic form of neurodegenerative disease, among others. Can I answer your question?
Thank you.
But following up on that though, is it known why some people never get Parkinson's, Alzheimer's, cancer, heart disease, diabetes?
Those are the blessed ones. So yes, of course, if you think about it, Our Lady of 122.5-year-old, she perhaps didn't have these diseases until quite late. So yes, certain people that are blessed, meaning they don't have this disease until very late stage. Yeah, so it's interesting to ask that question. There's all these studies, because I'm in the area of aging, so my colleagues have studied centenarian studies, meaning they collected individuals who live over the age of 100 years old and asked the question, like they asked this lady, what did they do that I'm not doing? Or is there anything we can learn? It turns out these people, they are truly genetically blessed. From a lifestyle standpoint, they may be, not all of them, they may not exercise as much, they may smoke like this lady does, but I don't, just want to make sure I don't recommend people not to exercise or not smoke, because these are individuals that are genetically blessed. I don't consider myself so much blessed, so I need to, look into these healthy lifestyles. Yes?
Are there any medications currently Are there any medications currently that can protect the integrity of the mitochondria, or any medication in the pipe?
Yeah, so in that context, we asked the question about are there potential medications that can, let's say, delay aging or something. Right now, there are certain medications that one can potentially help aging aspect in animals. But there's one key medication right now that is being recruited to study by the National Institute of Health and other private foundations. This medicine is called metformin, which is related to diabetes control. It's a medication that we have for a very long time, so we consider this medication old and reliable. So that the study, there are previously certain retrospective studies suggesting this might be helpful, but the true prospective studies are just beginning. So we would all know in a few years whether this is actually can delay aging or reduce the disease of aging. But it's very important to know this is the only medication that is currently in clinical trial. It's affecting mitochondria.
Wow, metformin for mitochondria?
Yes.
I didn't know that. In the back. Oh, no.
I would like to know, once you do have the Alzheimer, is there medication for you?
What this lady asked, if once you diagnosed to have Alzheimer's disease, are there medications for the patient? The answer is yes. So just until a few years ago, we have two medications that have been approved by the Food and Drug Administration, FDA. So one is the cholinesterase inhibitor, one is called memantine, which we routinely give it to patients. Their effect is okay, but not too strong. Most recently, there are 3 medications that have been approved by the food and the FDA, so those can delay the progression of Alzheimer's disease. Not too much, but it offers a great hope to, yes.
Slow it down a little bit. Yes.
I'm curious why it appears to take so. I'm curious why it appears to take so long to diagnose somebody with Alzheimer's.
Well, the process of diagnosing one disease, why it takes so long for any diseases, is a good question. Obviously, that has something to do with sometimes availability of clinicians that are properly trained in this field. For example, cognitive impairment traditionally is not a field that a lot of physicians would be interested in going to because it is financially it's not very lucrative, and then there were lack of treatment, effective treatment that can reverse the disease. Also, patient, you have a very long time taking care of this patient. Each patient and their families are in a kind of stress. You need to provide a lot of support for these patients. So that just says it sometimes takes long to diagnose these patients. Sometimes it's easy, sometimes it's harder. Yeah.
I'd like to know how you remove the damaged mitochondrial DNA, and also how do you measure the success of that procedure?
Yeah, so how do we remove, we mentioned, let me just see. So when we have a cube, so this has 2 components. One is the total mitochondria, That is involved in pink-Parkin pathway. So when we have this process known as mitophagy, meaning mitochondrial autophagy, so that ultimately remove these damaged mitochondria, that in a research setting, you can do multiple different markers, and it's quite routine nowadays. And then with respect to particularly mitochondria DNA, so this is technically much harder, but we can do these sophisticated techniques like RT-PCR, and then among others. Yeah.
Actually, the question was just that. Okay. Dr. Guo, any concluding comments for us?
I just wanted to say I'm delighted to be here sharing my research and clinical insight to these, thank you, to aging and these diseases that currently has no cure, but together potentially we can work to make the world a better place by finding cures and provide better clinical care. If you would like to be supporting our research, please contact us.
Well, you are making the world a better place. Dr. Ming Guo.